Hello students. Today's video lecture is on about genome editing techniques. But first we know must about the genome editing. What is genome editing? Genome editing or genome engineering is a type of genetic engineering in which DNA is inserted, deleted, modified, or replaced in the genome of a living organism. Unlike every genetic engineering technique that randomly inserts genetic material into a host genome, genome editing targets the insertions to site specific locations. Okay, now, as of 2015, four families of engineered nucleases were used, that is, meganucleases, ring finger nuclease, transcription activator like effector based nuclease, and the clustered regularly interspaced short palindromic repeat system. Nine genome editors were available as of 2017. All these classes of these enzymes, that is, zinc finger nuclease, TALENS, and meganucleases, and etc., are selected by Nature Methods as the 2011 Methods of the Year. The CRISPR Case9 system was selected by Science as 2015 Breakthrough of the Year. The first major class or technique in genome editing is meganuclease. The steps which are followed in this technique is number one is recognition, number second step is binding, third step is cleavage, fourth repair, and five outcome. Let us discuss the complete process of meganuclease. The meganuclease process involves the use of enzymes known as homing endonucleases, which are capable of recognizing and cleaving specific DNA sequences. These enzymes, often likened to molecular scissors, bind to their target sequences through complementary base pairing, ensuring precise recognition. Once bound, the new meganucleus cuts the DNA at specific positions within the target sequence, generating a double strand break. Subsequently, the cell's repair mechanism come into play either through non-homologous end joining or homologous direct repair. Non-homologous end joining can lead to the introduction of mutations at the cleavage site, while homologous direct repair allows for precise editing of the DNA sequence by incorporating desired changes. This process enables targeted genome editing, offering immense potential for applications in gene therapy, biotechnology, and basic research. The second technique is zinc finger nuclease. Is highly specific genomic scissor consists of two functional domains, a DNA which is binding domain and our second DNA is a cleaving domain comprises of nuclease domain of FOK1. Certainly, the zinc finger nucleases process involves the use of designer proteins to target specific DNA sequences for editing. Here is a brief explanation along with a simple diagram. The first step is zinc finger proteins. These are the proteins engineered to bind to specific DNA sequences. Each zinc finger module typically binds to three base pairs of DNA. After this, they attach to the zinc fingers proteins in a nuclease domain, such as FOKI, which is capable of cutting the DNA at a specific site. The ZFN binds to its target DNA sequences via the zinc finger domain. Once bound, the nucleus domain cleaves the DNA at a precise location within the target sequences. The fourth step is DNA repair mechanism. After the third step completed, the DNA is cleaved. A cellular DNA repair mechanism come into play. These mechanisms include non-homologous and joining, or homologous direct repair. After this, depending on the repair mechanism utilized, the DNA sequences may be edited, and non-homologous end joining often results in random insertions or deletions, while homologous direct repair allows for precise insertion of desired sequences. Now, the third technique is transcription activator like effector nuclease, which is a molecular tool designed for precise genome editing. And the steps are: number one, design TALEN; number two, binding to target DNA; third is dimerization of fork domain; number four, double strand break formation; five is DNA repair mechanism; six is functional consequences. So TALENs consists of two main components, which is a customizable DNA binding domain derived from transcription activator like effectors, and a nuclease domain from the FOKL endonuclease. Once designed, TALENs are engineered to bind to specific DNA sequences in the genome, where they induce double strand breaks through the dimerization of their fork domains. Subsequently, cellular DNA repair mechanisms come into play, primarily non-homologous end joining or homologous direct repair. Non-homologous end joining often results in small insertions or deletions at the break site, leading to gene disruption or knockout. On the other hand, homologous direct repair allow for precise editing when a repair template such as donor DNA is provided. Through this process, TALENS enables target modifications to be introduced into the genome, offering opportunities for gene correction, knockout, or insertion with high specificity and efficiency. The last technique used in genome editing is CRISP or Cas9 system, which is a powerful tool for genome editing that originates from bacterial and archaeal immune system. It begins with the acquisition of spacer sequences from invading viruses or plasmids, which are incorporated in the bacterial or archaeal genome within CRISPR arrays. These spacer sequences are then transcribed and processed into mature CRISPR RNAs by Cas proteins. The cRNAs, along with the trans-activating cRNAs, form a complex that guides the Cas9 protein to specific target sequences in the genome. Upon binding to its target DNA, Cas9 induces a double-strand breaks, which is then repaired by cellular DNA repair mechanism. Primary non-homologous end joinings or homologous direct repair, depending on the repair outcome. CRISPR Cas9 can induce precise modifications such as gene knockout, gene correction, or gene insertion, offering unprecedented opportunities for genetic manipulation in a wide range of organisms.